Welcome everybody to this webinar organized by EURO-NMD in collaboration with the European Reference Network for Rare Neurological Disorders and the European Academy of Neurology. We are honored that Professor Eduardo Tizzano has accepted to talk about SMA genetics in the therapeutic and newborn screening era today. Is a pediatrician and a medical geneticist and the director of the Department of Clinical and Molecular Genetics at the Vall Hospital in Barcelona, Spain. SMA is not the only area of expertise of Professor Tizzano, who's a very active academic and researcher at national and international level, and with different neuromuscular and genetic centers collaborations and participating as PI in several clinical trials, and also having professor tasks in different educational initiatives. It has wide liaison with regional, national, international patients and scientific support groups. And I could go on and on because the number of his titles are many and the connections to the RNs are multiple, as you see in his opening slide. We are honored that he also collaborates with us, the European Reference Network for Neuromuscular Diseases. And I'm going to hand over the floor to Eduardo and thank him very much for his kind participation.
So thank you, Antonio. It's a really pleasure just to share with you those last ideas and progress in the SMA genetics, specifically because we have a therapeutic and newborn screening era, and what is a traditional on the knowledge of SMA genetics, little by little is getting importance just to get all these new alternatives to the clinical practice, and particularly, for opening windows and new ways for research about the genetics of SMA and the genetic aspect of every patient with SMA. So I always put the picture of my team at the beginning because usually I never have time just to make the acknowledgement at the end. So my alternative is that you can now have a presentation or an introduction of my team, which I just really am very grateful for the everyday, impulse and just the desire to progress and doing things together in a tertiary hospital as a multidisciplinary team. So these are my disclosures. And just briefly, most of you know what is spinal muscular atrophy. My fair definition is that it's a motor neuron disorder which causes different degrees of weakness, mainly involuntary and other autonomous muscles. What is important here, you see that there picture, some aspect.
Sorry to interrupt, but we had a cut. Would you mind to restart the presentation of this slide, please?
Oh, yes. Can I do now, Antonio?
Yes, please go ahead.
Okay. This slide in which I define what is a spinal muscular atrophy, because most of you, even though most of you just know what is the disease, is a motor neuron disorder which causes different degrees of weakness, mainly involuntary and other autonomous muscles. In the other side of the picture, you see different aspects of the motor neuron and muscle pathology in the disease. And what is that we have neonates with severe SMA have up to 50% less motor neurons. And pathology also includes not only motor neurons, but also axons, neuromuscular junction, and muscle. This is more or less the milestones that happens in the spinal muscular atrophy during the last, we can say, perhaps 100 years or more than 150 years. And what we see here is that from the initial description of the disease by Wernicke and Hoffman, We have a clinical diagnosis and eponymous group classification in the 50s of 1950, between 1980, and started the genetic era with the cloning and characterization of the SMA gene of for spinal as a causative of spinal muscular atrophy. So genotype-phenotype correlation started in the studies, and 5 years later, we have the opportunity just to have animal models available and starting the era of the translational research in which we can test preclinical studies about the therapeutic of the disease. And was only 10 years after, practically, that the initiation of clinical trials was started in patients. So, and 5 years later, we have the possibility just to have the approval of nusinersen as an SMN2 antisense tailored therapy. In 2019, we have the FDA approval of Zolgensma as the first gene therapy with SMN1. And later, in 2020, we have the approval by the EMA and the approval of FDA of risdiplam, an SMN2 modulator. And now the EMA is also approved of risdiplam. And of course, we have more to follow in the progress of the disease. So with these milestones, I want to show you also that is a growing interest in SMA by scientists, researchers, and healthcare practitioners. And started what I mentioned with the genetic decade, in which the genes were most of the time really paying the attention of the researchers, the translational research era, and we started with the therapeutic era, in which we have more than 500 papers in 2019, arriving to 700 in 2020, and during the last 3 years, the number of papers had never just got down between 715, 677, and 706. So more or less, is getting one of the diseases in which a lot of attention is put in by researchers and the scientists in general. The disease and its experience. The primary manifestation of the disease is the weakening of voluntary muscles, but the experience of patient is dominated by downstream complications. Just means that we see a neurologist for the first attempt to see what happened with the weakness of the muscles, but we need also a pulmonologist for compromised respiration. There is impaired nutrition, so we'll need a nutritionist. We have an orthopedic specialist that will take care of the deformities and, of course, scoliosis that appears. And the limited functional ability needs a good physiotherapist program and team. So that means that this is a disease, even it's a disease in which neurologists are involved, it's a multidisciplinary team that have to be in the follow-up of this. Traditionally, these are the type of patients that we see in the clinic. This is type 1 disease. Patients never reach the ability to sit, the sitters and the walkers. We see type 1 disease is non-sitters, type 2 disease can be sitters, or type 3 are walkers, but they can lose the ability to walk, that can become sitters, and type 4 disease that most of these patients are walkers for a long time. And this is represented here in these Madonnas, in which we see the 3 different Madonnas from Raffaello. If we see lying down, no sitter, Bambino Gesù, a sitter, Bambino Gesù, and a walker from Bambino Gesù. From the etiology of SMA, it's a disease of 2 genes. Just really speaking about the genetic of the disease, the determinant gene SMN1, in which biallelic pathogenic variants, most of them deletions, are detected in affected patients and confirm the disease. The absence of these 2 just really is remarkable and straightforward for the diagnosis. I always speak to the patients, and particularly to the children and adolescents with SMA, that SMN1 is like a powerful car, like represented in this Ferrari, and that this Ferrari can be absent, can have 1 copy of the Ferrari, but with the point mutation. That means that the wheel is not there and the absent in the other, or you can have always, the copies of the a complementary gene, in this case represented by this small car, in which the number of copies varies from 1 to 6, and this homologous copy is present in all the patients. Most of the cases have 2, 3, or 4 copies of SMN2. With this in mind, we know that the one is the determinant, the absence of SMN1 is the determinant of the disease, and the number of copies of SMN2 is known now as the first modifier or genetic modifier of the disease. Speaking about the genotypes, we have in the left, you see here what is happening in the general population. With 90% of cases, we have people with one SMN1 and one SMN2 in each chromosome, in each of the alleles. And there is a 5 to 10% of persons that have absence of the SMN2, but the presence of SMN1 that makes them to be non-symptomatic and they don't have any problem. Both genes are located in the long arm of the chromosome 5. That's the reason that this is also known as 5q SMA. For the other side, we have the SMA carriers that have a prevalence of 2% in the population. And we have two types of carriers. One carrier that has the SMN1 traditionally in one allele with absence in the other one. But there is a few percentage of carriers that have two SMN1 in cis in one chromosome, and that can be also transmit a zero allele to the descendants. So this is important because sometimes you can study for carrier studies, not only the presence of one copy of the SMN1 can discard, and you have always a residual risk that can be two SMN1 copies in one chromosome. Patients with this, also there are, of course, there are some point mutations in the different alleles, and that can cause also the disease, and also they can be a carrier of the point mutations. Regarding the patients with SMA, there's an incidence of 1 in 6,000 and 1 in 2,000 live births, and there are no SMN genes with a prevalence of 0%. That means that we will see after, when we see the function of both genes, that even a small amount of SMN protein is needed in every cell just to survive. So the SMN2 most important difference with SMN1 is a CT transition at the position 840 in exon 7 of the SMN2. When we see in the powerful CAR, this position is cut by the cytosine, and in the SMN2 is a thymine. So that means that the TTC here, that is a codon here of TTC, just codifies for phenylalanine, and TTT also codifies for phenylalanine. However, that's considered as a silent change that in certain mode speaks because predict the same amino acid sequence but alter the recognition of exon 7 to be included in the messenger RNA. We are going to elaborate a little bit most concept. The presence of the C in SMN1 makes the production of an exonic splicing enhancer that is becoming very powerful to attract the proteins that favor the inclusion of exon 7. And on the other side, the SMN2, the presence of a T, just produce an exonic splicing silencer, and this exonic silencer creates attraction to negative proteins that hides the exon 7 and becomes not offered to the machinery of the splicing. That means that most of the transcript from exon 7 of the SMN2 is not present. And of course, in the SMN1, we have the presence of the complete SMN1 and the complete protein that forms an stable oligomer in the SMN complex. The SMN protein is a multifunctional protein expressed in all cell types, although motor neurons are more sensitive to its decrease. It has 294 amino acids and interacts with other proteins in a complex that plays an essential role in the assembly of splicing proteins. And it has also another role as an axonal transport and maintenance of the neuromuscular junction. For all these functions, probably come together to make the motor neuron most sensitive to the decrease of the amount of protein SMA. What happened with the SMN2? The presence of a T just causes splicing error in most of the transcript, and there is an unstable protein that we call SMN delta 7 that does not form a stable oligomer and is rapidly degraded. What happened, particularly when SMN1 is missing, and particularly in the spinal cord, SMN2 tries to compensate and can produce more of the complete protein. However, the amount of protein that is produced by the SMN2 never reach the amount of protein that is produced by SMN1. So the first concept here is that SMA is produced by less amount of SMN protein, not because of the absence of the SMN protein, because the absence of SMN protein is not compatible with life. So we have the first alternative that we can just envisage from therapeutics, that is just try to see or to take advantage of the SMN2 that is present in all the patients, just to make some therapeutic alternatives. Regarding the genetic diagnosis, when we have a patient with clinical suspicion of SMA, we perform a genetic test. The golden standard for this genetic test is MLPA, that is for multiple ligation-dependent probe amplification, a specific quantitative test for SMN1 and SMN2. And you see here that most of the patients, in this case, 95% of the cases, they have absence of SMN1, and we see here the absence of the copies of SMN1. That's biallelic absence of SMN1, just really confirm the diagnosis of SMA. There can be other situations in we have the suspicion of SMA and appears with one copy of the SMN1 or two copies of the SMN1. These are the special cases that should be studied by sequencing, including also false negative in the neonatal screening that looks only for absence of both SMN1 copies. And you see that we have to consider other diagnoses in most of the cases that they have two copies. But however, when exists consanguinity, is it possible that two rare point mutations are really shared by the parents and they will cause the homozygous alteration of the SMA protein, or SMA gene and protein. Of course, there are pathogenic variant reported in SMN1 in SMA patient. Almost 100 point pathogenic variants were being reported so far, and most of them are located in exon 3 and exon 6. However, we have to see that everything can happen, and depending on the different populations, we can have different type of frequency of mutations. For example, in our population in Spain, we have two main point mutation. In this case, 50% of our cases with point mutation have a deletion of 4 bases in the exon 3, and 10% of the cases is a duplication in the exon 6. It's an 11 base pair duplication in exon 6. So this is something that you have to take into account, that in the past, these 5% of cases, they were not totally well diagnosed, and now with the therapeutic scenario, they need to be diagnosed and confirm that they are SMA patients, just to these patients can have access to the therapeutic options. So SMA genetic diagnosis is not always straightforward. We can see there are things that happen with SMN1 and things that happen with SMN2. With SMN1, what is important, as I mentioned before, is that it's mandatory to have both SMN1 alleles characterized, and novel point mutations that are described need to be confirmed that they are in SMN1 or in SMN2. The real mutations are in SMN1 because point mutation in SMN2 may affect the function of SMN2, but not really be determinant of the SMA by alteration of SMN1. And patients with two copies of SMN1, as I mentioned before, and are consanguineous or with a high clinical suspicion of SMA should be studied by SMN1 sequencing. One note of attention that in some patients, one allele is deleted and another is present, one copy of SMN1, and the second allele cannot be characterized as mutated. We sequence it from the beginning to the end, and we are not able to find any specific point mutation or point pathogenic variant. What is happening with these cases? These cases may be other neuromuscular or motor neuron disorder that may be concomitant carrier of SMA. The possibility to be a carrier of SMA is 1 in 40, 1 in 60 of the general populations. That means that it is likely that sometimes we can have two problems concomitant, just as another rare motor neuron disorder that has no SMN1 homozygous deletion, but this has by chance can be a carrier of the disease. What we need to do in these cases, we will need to do a clinical examination very, very, very deep, do additional genetic testing, including NGS of the SMN region, in order to see if there is some intronic mutation or some rare mutation that cannot really catch it with the sequencing, standard sequencing process. and to perform sometimes an exome analysis that can reach all or a panel of motor neuron and neuromuscular disorders that can be performed to better categorize these cases. So I think that this is a challenge when we have a patient that we suspect SMA, we have one copy and we sequence the patient and we are not able to get the mutations because we don't know for sure if this patient will be another disease or we are just not able to detect the SMN1 mutation. And we are losing in this patient the possibility of a possible treatment. Just going to the SMN2 copies are really modifier of the disease. And in this case, the MLPA can also make a particular analysis of copies of SMN2 at the same time that SMN1. And in practice, most of the cases with type 0 disease, that is a more congenital form, have one SMN2 copy. Most cases with typical type 1 disease have two SMN2 copies, and patients that are sitters and walkers, they have between 3 and 4 copies of SMN2 in most of them. Every copy of SMN2 is able to produce a certain amount of SMN protein, and that's explained that patients with more copies of SMN2 have a more milder phenotype in comparison with patients with 1 or 2 copies of SMN2. And just doing a meta-analysis of more than 3,000 cases from the literature, we see that in overall, SMN2 copy number is a good predictor of the SMA type because most of the cases with type 1 disease have 1 or 2 copies. Most of the cases with type 2 and type 3 disease have 3 and 4 copies. But there are also some better than expected patients that they have type 2 or type 3 disease with 1 or 2 SMN2 copies. And there are others that are worse than expected, that they have 4 or 5 copies of SMN2, but they have type 1 or type 2 disease. So that means that we have what we call phenotypic discordances between the SMN2 copy number and the phenotype that present the patients. The reason for these discrepancies in SMN2 are particularly can be technical or biological. Usually the technical reasons overestimates the number of SMN2 copies, and the biological reason is observed in cases with less number of SMN2 copies than the expected according to the phenotype. And just to enumerate some of the recent discrepancies that are technical, they can be first an erroneous information or patient's misclassification, there is poor quality or low DNA concentration, the interpretation and control issues regarding SMN2 copy determination that are not good, and there could be variants in the regions of hybridization of primers of the probes, of primers or probes that are really in the MLPA or other genetic testing for SMA. Regarding that, a report of 2019 that genetic testing was repeated in 20 patients with SMA, and the second test used with MLPA, and the first time there was different other methods, and there was a discrepancy even arriving at 45%. So that means the overestimation of SMN2 copy is more frequent and will result in observing a phenotype worse than expected, but most of these reasons are technical. And when you have a patient that have more copies and the phenotype is really severe, you have to repeat the test, perhaps in another laboratory, in which you can just really have the confirmation that that is the number of copies that really SMN2 copies that the patient have. Regarding biological reasons, we can talk about hypermethylation of SMN2 copies. We can do negative modifiers that can have cryptic mutation or partial deletion of the SMN2. The presence of positive modifiers, we are talking a little bit later about that, that makes, that the inclusion of exon 7 is improved. It's not like SMN1, but it's between the typical SMN2 and the SMN1. That means that the patient has a milder phenotypes. Regarding the SMN2 structure, sometimes there could be hybrid genes or partial deletions that makes things a little bit complicated, and other modifying factors that could be epigenetic, environmental, that we don't know. And of course, it is important that sometimes the clinical evolution of the patients make a reassessment of the SMA type that had been put in the beginning. So we have all these reasons they have to take into account for the discrepancies. And I want to mention that the CT transition that is present in the exon 7, that is in the position A40, is not the only difference that is between SMN1 and SMN2. There is at least 15 paralogous sequence variants that differentiate SMN1 and SMN2 and is located just in exon 8, exon 7, intron 7, and intron 6. And of course, makes the condition a little bit more complex with difference. It's not that only the CT transition that makes specifically the definition of what is an SMN1 or what is an SMN2 gene, but also difference that makes hybrids just to be performed. What are the hybrid SMN genes? We have usually patients that have the SMN2 complete or the SMN2 from exon 1 to 7 of the SMN2 and exon 8 of the SMN1. This is an hybrid gene. Most of these hybrid genes, because the exon 8 is not totally included in the messenger RNA and in the protein. Of course, we don't know yet the alternative, but most of these patients probably have the same evolution of patients with complete deletion of the SMN1. But we have been studying more in detail, and we see that there are not only is the presence of the exon 8, as we see here, that is present in the patients. So starting from pure SMN1 and pure SMN2, we can have different type of hybrids just with the conservation of the exon 8 of the SMN1 and other different paralogous sequence variants of the SMN1, or you can have also hybrids that are most of this part are related to SMN2, and they have some nucleotides from the SMN1. The real implication of these hybrid genes is not yet known, but it seems that perhaps these patients that had hybrid genes with parts of SMN1 in comparison with patients that have a pure copy of the SMN2, probably in these cases, the evolution of the cases can be just really a little bit milder. But still, we need to work and to see more cases and to study more in deep the different hybrid that is present in the population of SMA patients, just in order to get a categorical conclusion on that. These are only preliminary results, and we hope that in the future we are going to be able just to know more in deep all the structure of the hybrid genes when the hybrid gene is present in most of the patients. And of course, mentioning particular variants in the SMN2, there is a particular variant in exon 7 that is a change of the predicted change of glycine by arginine, in which increase the 20% inclusion of exon 7. And there is more complete protein and less severe phenotype. A patient with 2 copies of SMN2 traditional will develop typical type 1 disease. But the patients with one of these copies with SMN2 with this variant will develop type 2 disease. That means they will become a sitter. And what is really, really amazing is that when you have 2 copies of SMN2 and both copies have the variant, the patient is a walker. Actually, we have found in our casuistic that around 25% of the better than expected cases are due to the presence of this variant. As we see here, we have tested SMN2 copies in patients with type 1 disease, patients with type 2 disease, and patients with type 3 disease. And we see that the patients with 2 copies, type 2 disease, and 2 copies of the SMN2, 17% of these have this variant. And the patients with type 3 disease with 2 copies, practically more than half of the patients with type 3 disease and 2 copies of the SMN2 have this variant. And what is really important to see that this variant is never present in patients with type 1 disease. That means that was the first proof that really increasing the amount of RNA and protein complete in patients can really have some kind of therapeutic effect. That means that this is a therapeutic gene. We can call this a therapeutic gene in the pre-treatment era. And of course, we have to think in mind that we have to go beyond the copy number variants in patients with SMA. The copy number is only the tip of the iceberg, and we have to see that it's not only the quantity but also the quality of the SMN2, in which what we have to deal with rare SMN2 variants, the hybrid structure that I mentioned before, other single nucleotide variants, the methylation status of these different genes, and of course we can talk about other modifier genes or global genomic interaction that also can make some effect in the phenotype. And talking that, the first proof that perhaps there will be genomic influence besides the SMN locus is the presence of haploidectic and siblings with different evolution. When we see a sibling that is affected in a wheelchair, and we test the sibling and see that there's an SMN1 homozygous absence with a specific number of SMN2 copies, we test a sibling that can be even walking or walking with some difficulties, uh, and we test and they have exactly the same homozygous deletion with the same number of SMN2 copies. And sequencing of this SMN2 that doesn't mean any difference between one sibling and another sibling. That means that there will be other genomic factors that may influence the final phenotype. And see here we have a list of representative cases with a female less affected, a male less affected, or female less affected also here when they are sister or when they are brothers. So, and the number of copies is the same. And look here, we have a Walker patients with 3 copies, and we have a Walker patients almost normal with 3 copies. And we have here a patient that can have type 2 disease, and the sibling can have type 3 disease. So all should be studied. And of course, every sibling with SMA needs to be studied in order just to get the approach of the genetic characterization of these patients. In summary, we want to put a continuous spectrum of phenotypes in SMA when all the patients have SMN1 absent or pathogenic variants, and the different SMA type depends on the number of copies and the amount of SMN protein, and the SMA manifestations will be the more severe cases to less severe cases. And of course, there will be positive modifiers, as I showed you before, that can make the phenotype less severe, or can be negative modifiers, most of them mostly under study, but not really totally categorized yet, that can make the phenotype more severe. So the SMA knowledge of genetics leads to 2 main therapeutic alternatives. On one side, the modification of the SMN2 splicing, that is increasing the production of complete SMN protein from SMN2, taking advantage that the fact that it's present in all patients, as we see here, we have 2 different molecules, an antisense oligonucleotide like nusinersen, and a small molecule that make an oral risdiplam. So on the other is the introduction of a version of the SMN1 gene that is absent in the patients via the SMN1 cDNA of the adeno-associated virus 9. And just to mention that we cannot put, we don't put exactly the complete gene in the adeno-associated virus 9 because it only reach only 5 kb. So we need to use the cDNA that is exon plus exon plus exon plus exon. So that means that the effect of this antisense oligonucleotide and small molecules like risdiplam are not affecting the cDNA that is present in the adeno-associated virus 9. And when you even make a combination of both, this really affect only the endogenous gene. These small molecules and oligonucleotide affect the endogenous gene, and this SMN1 cDNA start to be an autonomous replication. And we can see here, this is the production in a normal person in which most of the protein comes from the SMN1, and where it's absent of SMN1, we can have the SMN2 as the unique source of the protein, and we could use small molecules or antisense oligonucleotide to increase the amount of complete protein. On the other side, we can have the viral vector containing the SMN1 cDNA that goes into the cells, and the vectors has a cDNA forms that's and circular episome with autonomous replication, And this episome, if the cell is not divided, will remain in the nucleus, but not in the incorporated in the genome. So we have these episomes that are autonomous and produce SMN protein, and also we have the possibility to improve the SMN2 that is endogenous in these cases. I'm talking about the different therapeutics. We had been initially treating patients with manifesting the disease, and that's we can say that are post-symptomatic patients. But now exists because of the different studies and the clinical trials. Here I show you results, for example, of a nurture trial in pre-symptomatic patients. In blue, patients with 2 copies. In green, patients with 3 copies treated pre-symptomatically in comparison with patients with 2 copies treated post-symptomatically. That difference makes that when you treat pre-symptomatically, you have a therapeutic window of better response and better efficacy in these cases. And that's the pre-symptomatic phase in which now there is the possibility to get newborn screening to catch practically 95% of these cases at birth and makes a therapeutic decision as quick as possible in order to treat this patient and have the better evolution. But first, we have to talk a little bit about what is considered really pre-symptomatic. We started with a patient that can be clinical silent, and there could be some prodromic symptoms, and after became a phenoconversion, just doing a clinical manifestation of the disease. And this phenoconversion may appear also in a few days. We have to have some considerations regarding the definition of what is a pre-symptomatic SMA in genetically confirmed neonates. And here I have a list that the patient has not to have weakness or hypotonia. They have a presence of tendon reflexes, no tongue fasciculations, no diaphragmatic paradoxical breath, no bell-shaped thorax, no hypoxemia, no hypercapnia, no swallowing or feeding problems. They should have a CMAP more than 1.5 millivolts and a chopping tail score more than 40. With this one, we can say that this is really a truly pre-symptomatic patients. However, there are some patients that we have to look very carefully that can have prodromical symptoms that makes just that the phenoconversion is really imminent, and we should proceed as soon as possible in order to catch the patient in these phases as much as possible. And of course, when we have already a patient that have an homozygous deletion, homozygous absence of SMN1, we do that means that is positive in the screening. We do a confirmatory testing, and we proceed with the number of SMN2 copies. What happens when it's negative? It could be a false negative or a true negative. You need a true negative when they have the 2 copies of SMN1. On a false negative one, they have 1 copy of SMN1, and the other one can be an absence. That copy has a point mutation, and the SMN1 is an absent. So what do we have with these false negative cases is around 5% of the cases, and the clinical identification will appear when manifestation begin. So that means that these cases are lost in the neonatal screening. But we can test for 95% of the cases in which we can after see the SMN2 copies, and if there is 1 copy, it's probably type 0, and is most probably is not pre-symptomatic because type 0 disease is always present at birth. And we treat, there is consensus to treat patients with 2 and 3 copies, because with 2 copies has a high probability to develop type 1 disease, and patients with 2 copies have a high probability to develop particularly type 2 disease, and some cases also type 3 disease. But there is no a lot of discussion in treating these patients. But 4 copies that will develop probably types 3 and 4, and they will become symptomatic later in life. There is a doubt about treat, or just really discussion about treat immediately or treat at symptom sign onset, that we see, watch the patient and see what happens. There is no consensus. So treat or not to treat is the question for patients with 4 SMN2 copies. And here is a table in which we consider different factors regarding the treating of these patients pre-symptomatically with 4 copies, or when you treat when symptoms appear. You have advantage and disadvantage in all these cases, and we have to consider disease appearance and complications, the opportunity of treatment, the time to initiate therapy, the adverse events when continuous therapy, economic aspects that are also very important, the quality of life issues, and the parent and family expectations. So with all these things, still there is a discussion, and we still need a lot of biomarkers or several biomarkers that can help us to see the prediction of these patients with four copies. And what is important is that these four copies really represent really truly four copies, or they are not just a technical problem that overestimates the number of SMN2 copies. And of course, talking about neonatal screening, as neonatal screening is advanced and is expanding all over the world. And we see here from 2021, that was the map of the United States, and they were just in violet only the states that were screening for SMA. And now in 2024, the last January, we started just already in the States just to test all the cases by neonatal screening with SMA. That means that 100% of neonates with are tested in the United States. In Europe, the scenario is a little bit different because there are different health systems. And even though the newborn screening is expanded in Europe from 20% in 2021, now in 2024, we have more than 60% of neonates, and most of the cases are just established at national programs or pilot programs. So we expect be member of the SMA Newborn Screening Alliance. This European Alliance for Newborn Screening demands to national governments and authorities in Europe to immediately include the test for spinal muscular atrophy for all newborn children in national newborn screening programs. There is not more time to waste for babies born with SMA to start adequate treatment. The Alliance therefore calls on all decision makers in Europe to implement this essential health service in all European countries without any further delay by the year of 2025. Hopefully, we are reaching that in the next years, and we can have the possibility to treat all these patients. This is the things that says that no later than 2025. And of course, if we implement neonatal screening, the scenario of treatment will change. Now we have a few presymptomatic cases treated, and most of the cases are historical patients, and we have symptomatic newly diagnosed. If the newborn screening scenario appears, we will have most of the cases treated to be pre-symptomatic with a much more better evolution. We will have the false negative cases that they will appear the manifestation of the disease during the evolution of the disease. And we still have some historical patients that cannot be treated and they will reach the treatment after. So we are just really facing a totally different scenario in the future with the implementation of neonatal screening in most of the countries. And of course, there are challenges for newborn screening. There are challenges from the genetic, from the disease evolution, of the family aspect. From the genetic, we have to know that this is not a metabolic biochemical test. This is a genetic test to detect the absence of exon 7 of SMN1. You can have false negative at point mutation and false positive by rearrangement of rate variants, and the possibility to detect both patients and carriers. That will need a system that just can really be the cascade of cases that will come and we can reach that test to do genetic counseling will be impossible. Regarding the disease evolution, we have the predictive value of SMN2 copies and which patients to treat. As I mentioned, when you have a patient with 4 copies, the discussion is still open. There is need for other predictive markers as well as biomarkers of disease activity and treatment response. Neurofilament, that is a protein just liberated by neurons and particularly by axons, could be a promising biomarker, but still is on phase of research studies. And regarding the family, it is important to mention that the previous information, communications on diagnosis, and genetic counseling are really crucial in these cases. And there is a management of expectation and making decisions about therapy, because when the baby is born and they have the test already, they have to make a choice of the therapeutic decision in a few days. And most of these families don't know just what is really the the case of what is SMA. So that's the reason that we need to work on previous information and in a good communication of diagnosis team. Finally, I want to mention that we are moving now with the levels of prevention in SMA, that we have tertiary prevention, that is treat all patients with manifesting the disease, and the mainly influence of this tertiary prevention, treating these patients manifesting the disease, is in standard of care and evolving phenotypes. You know, they are changing the phenotypes that are appearing in the disease, and most of these cases reach the seated position and the walking position. But with complications like scoliosis and still some aspect of weakness or gastrointestinal problem or really swallowing problems. The secondary prevention is that treat all presymptomatic cases detected through newborn screening that may influence the burden and development of the disease. We have seen that the evolution of patients treated presymptomatically is much better than the patient treated postsymptomatically. So with the advances of genomic medicines, with the advances of genetic testing in the population, perhaps we are just reaching what is primary prevention, that is made genetic counseling in all the carriers detected through population screening to make reproductive choices. And what will be the influence of primary prevention? There will be influence in the incidence and the prevalence of the disease. A stay-home message, the first part regarding genetics and therapeutic, we know that SMA is a disease of two genes. SMN1 is a determinant gene that is absent or mutating in all patients, and SMN2 is a modifier gene that is present in all patients. SMN1 is homozygosally absent or deleted in 95% of the cases, and the remaining 5% of patients have deleted allele in one chromosome and a point pathogenic variant in the other. Few cases may be homozygous or compound heterozygous for point pathogenic variants. There is a strong correlation of SMN2 copies with phenotypes. The more the copies, the best the phenotype. But this correlation is not absolute, and some patients with two SMN2 copies may develop type 2 or type 3 disease instead of the expected type 1 disease. SMN2 variants may explain some cases of phenotypic discordance, and further the genetic predictors and new biomarkers should be investigated. Two main therapeutic strategies were developed based on the knowledge of SMA genetics. One is the modification of SMN2 splicing by nusinersen and risdiplam. Nusinersen is antisense oligonucleotide in the intron 7, and risdiplam is an oral compound that influences the splicing and the inclusion of exon 7, or SMN1 replacement by gene therapy that emerged as thyroid therapies in SMA patients. Regarding the second part of newborn screening is expanding in different countries. Early treatment and intervention improves the effectiveness of the therapy, showing or arresting the development of the disease. Newborn screening may detect 95% of neonates with SMA, and the remaining 5% of neonates are false negative for the screening and may develop symptoms before the treatment can be considered. Just the contrary with newborn screening, which most of the cases that you are going to catch, they are not developing symptoms yet, and you have a more wide therapeutic window. Cases with 2 or 3 copies are treated virtually everywhere. And this means in every country. However, neonates with 4 or more SMN2 copies represent a challenge to treat, and a consensus is difficult to achieve. It is crucial the availability of reliable laboratories to determine SMN2 copy number correctly. And of course, we need also the alternative that to have good biomarkers to see what is the evolution of the disease in these 4 copy cases. New insights in SMN2 variants, hybrid structure, and genomic influence may allow a better prediction of the evolution of SMA cases, and the phenotype correlation should be considered beyond SMN2 copies in this moment. Primary prevention emerges as a possibility to perform adequate genetic counseling and reduce the incidence of the disease. The detection of carriers in the general population is gaining consideration in some health systems. And for example, in Australia, Israel, Netherlands, they are doing some official programs and some pilot programs to study SMA carriers in the populations. For me, it's just the last slide, that the future can be different. We have been moving during the past in improving quality of life of these patients. Now we have the possibility with the three different treatments to stop or slow the disease course, particularly when we catch us in the early stages of the disease. And of course, we are arriving to the limit of the cure, perhaps in the future, with an arsenal of different therapeutic alternatives, not only one therapeutic. We have to consider that the treatment of the disease will be probably with more than one alternative, and the combination should be studied in the future in these patients to see and to improve the quality of life and the efficacy of these medications. So I hope that you have an idea, and I open to questions, so any doubts that have been just raised during my talk. And thank you very much for your attention.
Thank you, Professor Tizzano. It was a fabulous expose of the situation for a disease who had a tragic destiny and now is becoming a treatable one. I have promoted our chair of the SMA working group, Professor Jan Kirchner, to a panelist, and I would be very happy if he would be able to unmute himself and collaborate in this discussion. So there are some questions popping up in the Q&A. So someone asks, which one is the preferable treatment?
I know that there was, even you speak about genetics, they were always a question about which one will be the best treatment. So I don't think that there is a best treatment. The alternatives that we have three different treatments, the three are they are good individually, and they have different indications depending the age of the patient, the SMN2 copy numbers, the evolution of the patients. But I cannot say really which one is the therapeutic option. So I'm convinced that in the future we are going to face more than one therapeutic option. So just choosing one of the therapeutic option does mean that I'm just making a treason about my ideas that will be in the future. We can have some other approaches to treat the disease. And what is clear that when you catch this disease even very early in neonatal period, you can have patients that are already prodromic or just minimally or post-symptomatic, and the disease, even though the disease improves, whatever, you cannot reach a total cure of the disease. That means that we don't have yet the time and where, just when to treat these patients as best as possible, and with what else we have to help that good medications, that really tailoring medications to improve the patients. But there are a lot of several clinical trials that are ongoing, and I think that in the next years we are going to have a therapeutic arsenal that we can treat these patients with more than one solution. I don't know, Jan, if you want to comment on that question or you want some other comments on that?
Yes, I think Jan is going to comment. He raised his hand.
Yes, thank you. Eduardo was an excellent presentation. No, it was not directly on this. I just had another question. You mentioned the debate about treating pre-symptomatically patients from newborn screening with 4 SMN2 copies. Do you think in clinical routine care we might need to use more genetic markers to identify those who might need treatment, or do you think that's not realistic in the near future? That's one question. And the other aspect is, if we now start treatment very early, it might be very difficult in the future to have a genotype-phenotype correlation because they all behave differently due to the treatment. So probably this needs to be done on historic cases rather than on new cases. So these are my two questions.
Oh, the two questions are very interesting. Regarding the first, I think that now we are coming in a way in which when you mention, the routine clinic and the routine genetic diagnosis is really nothing in comparison with the price that they have the medications. So my idea is that we have to know as much as possible of the patients that we are going to treat. And if we have the possibility and the tools to characterize these patients, in the future, probably they will help us to make some much better conclusion about the prediction or the evolution of these cases. And that will partially answer your question, in which if we start to treat all the patients with 4 copies, we'll never know when just really will be the natural history of these cases. But we are moving in a transition now, and we have to collect as much as possible, these cases with 4 copies and to characterize what are the really the quality of these 4 SMN2 copies. And not only that, but also the possibility to get some other biomarkers that can help us, like, for example, doing a CMAP or doing some neurofilament and see what happens with other biomarkers that may appear in order to have a global picture of what's going on in each patient. What I'm convinced is that we have to be aware that we have to be as personalized as possible. You cannot say, okay, all patients with 4 copies we will treat and doesn't matter what happened. So I know that treatment decision is an urgent decision and it's very important, but we still are investigating these cases, and we need to learn a lot about these cases. And of course, treatment is not just really cheap. So when we put this patient in treatment, sometimes you don't know what will be just really even the genetic tests, they are not complete, and there are several doubts. So I think that we have to start with this battery of different studies that we do at the beginning of the treatment, and we will need just to know what will be the prediction on that after the treatment. It's a pity that we cannot follow the natural history, but still there are some countries that are not treating patients with 4 copies and see what will happen retrospectively or even prospectively in that countries to see what happened with the patient with 4 copies. But for sure that we have to change the way in which we see the disease. We need to do more deeper studies on what is ongoing in each patient and try to make conclusions that are more from individual point of view, having a general view of what's happening in all these patients.
So Yanis Demetriadis wanted to know your opinion about the role and place for carriers screening. So he's wondering if you think it will be generalized one day as it is becoming for newborn screening.
Now, the transition is that in the future, we are going to have genomic in the health public, and that means that every person in the country will have a genome study. And this study of genome that will give you a lot of clues, a lot of information about if you are carrier of severe autosomal recessive disease and you have to test your partner. And this also will come possible with SMA. Now, NGS methodologies can just really catch the deletion of the SMN1 gene. And of course, I hope that in the future we are going to know persons in the general population that are carriers, and you can make genetic counseling, and the reproductive issues should be discussed. And in the future, the incidence not only of SMA, but a lot of rare disorders, they are severe rare disorders, will just really be lower than is now. Because if we have to think that every rare disorder can have a treatment that is so expensive like SMA, there is not health system that can really sustain the fact of the economical issues on that. So doing neonatal screening is good money because the treatment of this patient as early as possible is better evolution. And doing carrier screening, not only for SMA, but only for other severe diseases in the future, just really will make a difference in the way in which we face all these cases that now appears to be in our clinics practically every day.
Okay, we have more questions, but I'm going to read the final one, if you don't mind, because we are approaching the end of the webinar. The primary prevention would mean eliminating the disease in the future, someone says. In which way are reproductive decisions or genetic cure, for example, by CRISPR-Cas9, discussed with the patient community and the activists? Should this be a broader discussion? Question mark.
Oh, yes, I think so. You know, the most important thing is that you have the tools to make the diagnosis. Once you know, to have the knowledge is power. Knowledge is power, and it's empowering of the patient. If I know that I have a problem with my genes and I can transmit my genes to another generation, I have the power and my autonomous decisions to do whatever I want. And this is important thing that we have to discuss in a forum and with all different authorities. Patients have to have the voice also to make their opinions. But the important thing is that we don't decide for the patients. We give the patients or the carriers the alternative to know. They can know or not to know, but if they want to know, they can do that and they can make decision about knowing that.
Okay, thank you. I want to thank everybody for attending this very interesting webinar. Thanks to Jan Kirchner for your participation as well. And thanks to Professor Tizzano for the excellent presentation. And we'll have many other opportunities to be back together and discuss these themes in the future, I'm sure.
Thank you very much. Thank you very much. Bye, Jan. Bye, Antonio.
Bye.
And to next time.
See you next time. Thank you.